Welcome to Revolution Against Evolution. I'm your host, Doug Sharp.
Your co-host, Rich Gear, here as well. And Doug, the chemistry major and DNA guy, I'm going to let him do some, well, I always say that, but then I always end up getting in there, getting my fair share in there.
Yeah.
But you talk about the RNA, the ridiculousness of the RNA world. What do we want to title this show, Doug?
Well, we're going to talk about the RNA world, which is a conjecture that was made, I don't know how many years ago, of how life could have come about by chance.
Now, did this come up, this RNA postulation? Because I know I've heard about this because the DNA thing is so implausible. They've had to make a workaround, or
well. You start with they started out with the Miller-Urey experiment,
right,
which is what had me fascinated back in my high school days, and I tried to extend that, and by combining amino acids together to try to form proteins. That's where I ran into my impasse. And that's how it got me started down this road. Now, because they had recognized that this impasse exists with the chemistry of proteins, then they come up with this conjecture that the origin of life actually started with RNA, and RNA is a little bit simpler than the DNA molecule,
right?
And so they figured that the RNA, you could they had some experiments where they were causing it to what they thought was self-replicating.
Okay.
And so they were looking at the chemistry of life, starting with RNA, and watching the chemistry work, and and they said, well, this sort of shows that the chemistry of life is possible under certain circumstances, and they were having it actually work in clay materials, and they were thinking that it was this could have been the start of the origin of life.
Okay.
Well, I was looking at this, and I said, first of all, you're still going to need your proteins, because without the proteins, you're not going to have the enzymes that you need to catalyze the RNA reaction.
So the same thing, you're in the same problem in a way with the RNA as you are with the DNA.
That's right. Yeah. Okay. It's all controlled by this whole manufacturing process that goes from DNA to RNA to proteins, and then the proteins are used to catalyze the formation of DNA polymerase, which is the proteins that is necessary for the formation of DNA.
So you need protein to get the DNA, to get the RNA, to get and you need that, but you need the DNA to get the proteins. Isn't it kind of a circular? It's like
it's a circular process.
DNA needs everything to work, right?
Yeah, you need the DNA to. And each of these steps along the way in the manufacture of life, the whole living system is a DNA to RNA to protein to amino acids to proteins the process. And so what is needed is an explanation from the evolutionists of how this whole massive process got started.
Now everyone's kind of familiar with the DNA double helix and connected with the sugars, right? Got the kind of like the ladder in between this double helix. And RNA, you said, is somewhat simpler. What is the difference between RNA and DNA exactly?
Well, RNA is a half strand.
It's a half strand. Okay.
Okay. So the DNA is a double helix, whereas the RNA is a half. It's just one strand of it.
And doesn't hook up another, because I got all confused back when I started talking back in the old biology class. They had trans RNA and they had all these different kinds of letters in front of the RNA, and I was getting kind of mixed up because there was so many different kinds of things. Let me
explain what those all are. Okay.
That would be good for the audience, I think.
The most important piece is called messenger RNA, which is m-RNA. And what the messenger RNA does is it's taking the code that comes off the DNA molecule, and it is something that tells what sequence of amino acid to put on the growing protein chain.
Okay.
And then there's the transfer RNA. There is a piece of RNA that coils up into a little ball, and it's a three-dimensional conformation of this that
Really?
gloms onto the piece of amino acid, and it adds it to the growing protein chain. And so there are little things called codons, which are three sequences of nucleotides on the RNA that connect to the messenger RNA like this, and they recognize it, and based upon these three codes, it has the correct transfer RNA that holds the amino acid that goes onto the protein chain. Okay, then you have r-RNA, which is ribosome RNA, and that's a little bit more complicated. This is the whole housing mechanism in which all this transfer process takes place. And so this holds it together. It opens it up and lets in the transfer RNA, and then as it's reading, it's sort of like the machine that you're having the computer code go through for the manufacturing process.
Wow.
And so this is.
It seems like the more you know, the more it seems highly implausible that any kind of non-intelligence, just things starting by chance. Highly implausible. I mean, I'm understating the case.
Right.
How ridiculous it is. But so there's three different kinds of RNAs. Then you're saying, right?
There are, yeah.
Okay.
Now I've got an article here that was taken off the CMI website, the Creation Ministries International.
Yeah, I was reading that
and you
earlier.
find this at creation.com, and there is a quotable quote by Karen Smith, which is a physical chemist, and he's well known for what he calls a bizarre theory that the first living organisms were clay minerals of the origin of life.
Right.
And so...
And there was a reason why they were thinking that, right? Because the DNA or the stuff kind of sticks to the clay or something like that? Or
he says, Professor Deemer says that amino acids in DNA, the building blocks for life, and phosphate, another essential ingredient, clung to the surfaces of clay particles in the volcanic pools.
Right.
The reason for that this is significant. It's been proposed that clay promotes interesting chemical reactions relating to the origin of life, he explained. However, in our experiments, the organic compounds became so strongly held to the clay particles they couldn't undergo any further chemical reactions. But he's still, and so it talks about the implausibility of the pre-vital nucleic acid, which means before life came about. And it says, if it's hard to imagine polypeptides or polysaccharides in primordial waters, it is harder still to imagine polynucleotides. And knowing the chemistry of this, the nucleotides are a little bit more complicated than amino acids. And so, but these are the building blocks of life. You need to explain nucleotides, and you need to explain amino acids, and they make up the lattice chain of DNA and the matching proteins that come out of it. And the problem is that the reagents that you need to make nucleotides conflict with the reagents that you need to make amino acids.
Oh boy, it's like...
And so you've got to now go over here and separate in this little area environment.
Well, Urey had that, Urey, they had the same kind of problems dealing with the amino acid. They had to separate what they made supposedly from the actual water or the primordial soup, which if it went back into it, it would just dissolve or assimilate back into it. So yeah,
and that's sort of what I found out when I was zonking amino acids to try to form proteins. The electrical spark that formed the amino acids in the first place. If you send it back through the process, it breaks it down.
Yeah.
Well,
yeah, because it seems to me like the basic building blocks are so tenuous and fragile on themselves unless they're already completely put together the way they meant to be. And in a certain thing, there's no possible way they could ever combine or even become created in the first place to combine with the other things needed to make life happen. As I say, the properties, inherent in matter do not lend themselves to life happening.
Yeah. Now, the writer of this article, and that would be,
excuse me,
I think it's Jonathan Sephardi. He talks about 19 different difficulties.
Yeah, I was reading some of those. I was kind of going, wow. I know Sephardi. How do you pronounce? How do you spell his name? S-A-R-F-A-T-I or something like that.
S-A-F-A-R-T-I or something
like that. He's a genius, though. I mean, the guy. He's written several books on this kind of stuff. But I started reading those, Doug, and I started getting my eyes were starting to glaze over. But it's really good.
If you understand what's going on chemically, this is.
And most people don't, Doug. So we have to break this down because I know I get probably more than maybe the average guy. You're a lot more than me, and most people are not going to understand. When he's writing these things, you're going, I'm taking it for granted. These are problems, but I'm not quite following everything, as I'm reading them.
Okay.
So maybe you can kind of explain them as you're going to, or go through a few of them as you're going to go. Are you going to explain them?
Yeah, I'm going to explain these as we go
go
along.
for it. Yeah.
The first is it's not even clear that the primitive Earth would have had generated and maintained organic molecules. And all that we can say is that there might have been pre-vital organic chemistry going on, at least in special locations. But the second, the high-energy precursors of purines and pyrimidines had to be produced in a sufficiently concentrated form. In other words, the problem with the big universe is that it has a tendency to dilute chemical reactions.
Yeah.
Okay. The third, the conditions must now have been right for reactions to give perceptible yields of at least two bases that could pair with each other. And so you got base number one, that there's four bases, just like baseball.
Yeah, right, yeah.
If you talk about DNA, you talk about base pairs.
Yeah.
And they count base pairs because they combine together in the DNA molecule.
It's like A and G or T or
adenine, thymine, guanine, and cytosine.
Cytosine, yeah.
And in RNA, there's a fifth one called uracil.
Really?
Yeah.
Interesting. I thought RNA was supposed to be simpler.
Well, RNA replaces one of the bases with the uracil.
Really?
And so I actually don't remember how that works, so don't ask
I will put them on the spot right now because I certainly don't understand it. There are probably chemists out there that probably do get it a lot better, but they're realizing just with some of the words, some of the problems that are going on here. So what are some of the other ones, Doug?
Well, these bases must have been separated from the confusing jumble of similar molecules that must have also been made, and the solutions must have been sufficiently concentrated. And so we have the problem of dilution of the stuff.
Yeah.
Your warm pond would have diluted the chemicals sufficiently so that they wouldn't have reacted.
Well, Doug, this is the same we talked about in other shows about how the very neat thing that needed for all living things is oxygen. is also the very same thing that's destructive for anything from being created.
Right,
I mean,
and water
water's even worse.
is an enemy for the formation of the peptide bond. And
Yeah.
so they have to figure out how to actually do it in a dry environment, and then if you have a dry environment, it doesn't help it at all.
That's
so.
why I laugh a lot of these people go, oh, Mars has water. All we need is water to show life could have happened. I said, I'm sorry. It takes a lot more than water to show life could have happened. You need it, but unless life is already working and functioning, it'll destroy it.
Yeah, H2O is, it's one of the simplest molecules that's out there.
Yeah.
But it's abundant, and they need it to have things in solution. But if you have things in solution, it disassociates.
Yeah, it destroys what, yeah.
Yeah, it's a problem, Fifth, in some other location, okay, so you have over here another reaction, for a formaldehyde concentration above a certain concentration must have been built up. And this accumulated formaldehyde would have been converted into sugars, and the word that they use was oligomerase.
Okay, that's a sugar?
Yeah, that's that's the process that converts formaldehyde to sugars.
Oh, okay.
Now, formaldehyde itself is a pretty nasty, caustic chemical.
Yeah.
And so it would have had to convert from formaldehyde to sugars. Seventh, somehow these sugars must have been separated and resolved to give a moderately good concentration of D-ribose. And this is another problem that would have had, is that they're right-handed and left-handed sugars.
Yes, right.
And there's actually 4 different conformations of sugars, but it only uses the D, the right-handed sugars
yeah.
In life. And so then they would have to figure out how to separate the bad sugar from the good sugar.
Or the one that doesn't work for you, yeah.
Right. And then bases and sugars must have then been able to come together. And then they would have been induced to react to make nucleosides. And there's no ways of bringing this out thermodynamically uphill reaction in aqueous solution. Purine nucleosides have been made by dry phase synthesis. Not even this method has been successful for condensing pyrimidine bases and ribose to give nucleosides. And so we're assembling the DNA molecule now, piece by piece.
And what he's saying is each piece doesn't work.
Yeah, this piece doesn't work. 10th, whatever the mode of joining base and sugar, they haven't figured that out yet. It had to have been between the correct nitrogen atom at the base and the correct carbon atom of the sugar. This junction will fit the pentose sugar as either the alpha or beta anomer of either the furanose or pyranose forms. Okay, what it means...
See what I mean, audience? I tell you why I was getting glazed eyes on this.
Yeah.
I'm not following all of this stuff. I mean, I'm getting the gist of some of this stuff, but it's pretty in depth there.
And in the experiment that they did in the dry phase, this is out without water, the synthesis only produced 8% of the time the correct structure. And so there's different ways that the base and the sugar would have joined. And if you don't get it right, it doesn't work. And so, and only 8% of the time, you get the right conformation.
Well, wouldn't they say that this 8% though, Doug? Well, over millions of years, there would have been enough of them to combine or something?
Well, you have to get it right all the time in the first synthesis. And so you're talking about hundreds or thousands of these coming together. And if you only get it right 8% of the time, the percentages get less and less for each step of the ladder that you can join up.
All right.
And so this is a real problem. And 11th, the phosphate. There's a phosphate involved in this. must now bend precedent at a reasonable concentration. You need the phosphate there. The concentrations in the ocean would have been very low, so they have to think about special situations like evaporating lagoons and such things.
So all these experiments, they've really every time they try it, even and failing, even though they're doing this,
Right.
they're artificially changing the way things really are in nature.
Right, they can't, they're trying to come up with a special situation that
They might have
actually
done
works.
this, you know. Okay.
And then the phosphate had been activated in some way so that, and this is energetically uphill, phosphorylation of the nucleoside is possible. So you have the sugars and the nucleotide and the phosphates have to come together in a correct manner, and and only the 5-hydroxyl of the ribose should be phosphorylated, which is there's 5 little joining mechanisms on the sugar, and there's only one of them that's correct for the phosphate. Okay.
So it gets even worse. Okay.
Yeah, so this is work. And then they haven't been able to work this. It says, and if you heat it, it gave the nucleoside, it goes to the 2 or 3 position rather than the 5% position as a major product. And so this again greatly decreases the possibility of this forming. Number 14, if it's not already activated, the nucleotides must now be activated, and the reasonably pure solution of it must be a reasonable concentration. And so chemically, now these things go through phases. If you're actually trying to synthesize this in the test tube, you have to, it goes through a bunch of steps to get the pure form of it.
Oh, I see. Okay.
And
is that for each of these steps, this kind of stuff? That you have to get all these.
Exactly. You have to get all these different steps.
Okay.
And then you have to have a suitable coupling agent that would have to be fed in such system. So there's a reagent that you need to make those things join together. And then the activated nucleotide, is number 15, must now be polymerized. In other words, it has to repeat itself, a whole bunch of different things. And this number 15 has 3 classes of problems. You know, this is probably beyond the scope of.
Yeah, I'm saying. I tell you, so yeah.
It's probably beyond the scope of this program because.
Yeah, we only got 5 minutes left on our show, Doug. Yeah. So you want to keep it going, but you can see, the audience can see how complicated and how insurmountable. Doug, it seems to me, unless you just had, there's an old thing, so a willing suspension of disbelief. No one would ever believe you could do this. Why would they waste millions of dollars?
And these are the
man hours to do.
These are taken from the notes of the chemists who proposed this sort of thing.
Yeah.
And they're coming into these difficulties, and as they were synthesizing the DNA molecule, and so. You can read all these problems with the number 15 here. And number 16, the physical chemical environment must at all times become suitable, like the pH, the temperature, and and the concentrations. And 17th, all reactions must take place well outside the ultraviolet sunlight.
So it's going to be some dark cave or something, or what?
Yeah, you'd have highly destructive effects on the nucleic acid molecule away from the radicals produced by the sunlight. And the 18th, unlike polypeptides, where you can easily imagine functions for imprecisely made products, genetic material must work rather well to have any use at all. Otherwise, it will quickly let slip any information that it has managed to accumulate. And 19th, what is required here is not some wild one-off freak of an event. It's not true to say it only happened once. The whole setup had to be maintained for perhaps millions of years, a reliable means of production of activated nucleotides at the least. And so here are 19 reasons given by the evolutionary chemists themselves, which they show why the problems that they have in creating life in the test tube. I came to the conclusion early on, years ago, seeing just a few of these things.
Yeah, I remember. Yeah, we talked about it on the show.
Yeah, I haven't even mentioned anything about the formation of amino acids and proteins.
No, we've done that before, but yeah, it's the same issues arise. This test of Dr. Chan, that was pretty good. I thought that was pretty amazing.
Yeah,
What do
you want to read a little bit of this?
I will. It's really pretty good. I was really impressed with this. Got a couple minutes left, so hopefully we can get through this. We should be able to. In 2005, I started my work as a college professor at University of Missouri. I was an ardent believer of Darwinian evolution at the time, believing that life came from non-life and complicated life from simple life. I had a 1 year break, i.e. without teaching obligations, so that I could set up my laboratory. 2006, I started teaching molecular biology using the molecular biology textbook by Robert Weaver, a popular molecular biology textbook. Okay, he says teaching is a lot different than being a student, but he says my main goal is to remember what my professor said in order to pass the exam. But now to teach the course myself, I need to pay attention to and understand the details of the subject. I observed that the molecular machinery is responsible for DNA replication, transcription, and translation in bacteria, archaea, and eukaryotes, which are the three domains of life, are not interchangeable, and a machinery that functions in one domain of life cannot be made to function in another by conceivable adjustments without killing the organism. So this directly contradicts with the expectation of the popular theory that eukaryotes have evolved from prokaryotes, bacteria and archaea. The only theory I had heard of by that time. This is because decades of studies of genetic information processing and reproduction of various organisms, except RNA viruses, have demonstrated that only DNA sequences can be directly copied and passed on from a parental cell to its daughter cells, that RNA and protein sequences are determined by the sequences of DNA and are indirectly copied and passed on by their being encoded within the DNA. In other words, he's saying there's no cross. You can't get from one type of domain of life to another. It's not going to happen, because for years I remember reading about that. The bacteria brought us into the more complicated stuff, and there's more to this testimony. We'll put it up there for you to read. But
so right at the outset, the evolution doesn't work at the foundational level, which is your chemical evolution of life, and if it doesn't work there, it certainly doesn't work when you get more complicated. Going down the road. It gets worse and worse. So we'll see you next time on Revolution Against Evolution.